Welcome to the zoology class. Fifth lesson in zoology, molecular genetics. In this lesson, we are going to study about genes in detail. What are the components of the genes? How informations are stored in the genes and how these informations are translated. So all these things we are going to study about in this lesson. At the molecular level, we are going to study the genes. What are the components, what are the elements, what are the atoms there in the nucleus, okay? And this is the structure of the DNA, deoxyribonucleic acids. In that, you can see the elements. What are the components you can see in the diagram, okay? Adenine, thymine, guanine, cytosine, then sugar molecule, phosphates. So these are all the components of DNA molecules. This is actually nucleotide sequence. In a DNA, many nucleotide sequence will be there. And each nucleotide sequence is actually a gene. Okay, so in this lesson, some of the things we are going to study about these things: genes, genome. Genes means a single nucleotide sequence in a DNA. Okay, so in a nucleic acids, a single nucleotide sequence is actually a gene. And what about the genome? Genome means it is a long sequence of nucleotide, and it is a complete set of hereditary informations of a organisms. Genome means, for example, you may studied about human genome. Human genome means so all the genes which are found in the human cells. So that is the study of all the genetic informations of the particular organism is actually the genome. Okay, genes means a single nucleotide sequence. Genome means the entire hereditary informations which are found in a particular organisms. And we are going to study about structure of the DNA, replication, transcription, translation. So these are all the three major processes: replication, transcription and translations. These processes we are going to study in this lesson. Okay, first, the DNA is converted into RNA. That process is transcription. And then RNA is converted into protein. Proteins are produced from RNA by the process of translation. Okay, this is transcription, translation. RNA is produced by the process of transcription, proteins are produced by the process of translation. And one more thing, replication is there. DNA is again producing DNA. DNA is producing two own copies. That is replication. Okay, replication means production of DNA by DNA. So these are the major processes which are going to study in this lesson: replication, transcription, translation. Okay, so this is the structure of the cell. Look at that. In a cell, at the center, what is there? Nucleus is there, and the nucleus, DNA. This is DNA is there. From the DNA, what is produced? RNA, mRNA is produced. So that process is actually transcription. The process of production of mRNA from DNA is transcription. And another one process, translation. From mRNA, what is produced? These are all proteins. Proteins are produced. So the diagram is exhibiting the three processes. Actually, replication, transcription, translation. This is the DNA. It produced more DNA by the process of replication. The DNA is producing RNA by the process of transcription, and the RNA producing proteins by the process of translation. Okay, next. Genes, the functional unit of inheritance. The unit which is responsible for the transmission of characters from parents to offspring. That is gene. Okay? Gene is the functional unit of inheritance. That is transmitting characters from parents to offsprings. That is responsible for our specific characters. Okay, first, Mental only introduced the concept of gene. The gene concept was introduced by Mental, but he didn't name it as gene. He named it only as a factor which is found in the nucleus that is responsible for transmission of characters. So that is only given by Mental, and he didn't name it as gene. The name gene is given by Johansen. He only named the genetic material as gene. Okay. Next, properties of the genes. What are the properties of the genes? In any organisms, the number of genes are more than the number of chromosomes. In any organisms, the number of genes will be more. For example, look at this. This is a diagram of chromosome. In one chromosome, this is a one chromosome, many genes will be there. Here one genes, here one genes. So many genes will be there. So from that we can understand which is more, number of chromosome is more or number of genes are more. Okay, in any organisms, number of genes are more than the number of chromosomes. And then genes are arranged in a linear pattern. Okay, the second point, it's a linear order. Genes are arranged in the chromosome in a linear order. One by one, it is arranged in a linear order. That is the second point. And locus. Genes are found in the specific position in the chromosomes. That is locus. Genes are found in the specific position in the chromosomes. Now look at that. This is one chromosome. In a chromosome, this is one locus, and here one locus. Locus means the location of gene in a particular place in the chromosome. Okay, so that is locus. So in this locus, the genes responsible for eye color is located. The genes responsible for eye color is located in this locus, and genes responsible for hair color is located in this locus. Okay, locus means it is the place, specific place of the gene. Okay, and then alleles. Genes exist in different allelic forms. Genes exist in alternating forms. So for example, here B, this is a gene B, capital B. What is the alternate form? The alternate form, this capital B is small b. So here two alleles are there, two alleles. Okay, one is dominant allele and another one is recessive allele. So genes may exist in several allelic forms. So here the genes are only in two forms, dominant form and recessive allele. Okay, in Pisum sativum, you know already, there are two alleles, capital T and small t. Capital T is a dominant allele and small t is a recessive allele. Okay, allele means that a different form of the gene, different form of the genes. Okay, now there are two hypotheses regarding this gene. So one scientist, he is saying that one gene is capable of producing one enzyme. One gene is producing one enzyme, or one gene is producing one protein. Now look at the diagram. This is the structure of the hemoglobin. This is the structure of the hemoglobin. It is made up of 4 polypeptides. This is beta polypeptide, beta 1, beta 2, and then alpha 1, alpha 2. 4 polypeptides are there. So they combine together to form a single protein, hemoglobin. Okay, so based on the first hypothesis, one gene is producing the entire protein or the entire enzyme. That is one gene, one enzyme hypothesis. Okay, that is studied in a mold, in a red-browed mold, Neurospora crassa. A scientist studied that one gene is controlling the production of one enzyme. But the second hypothesis is saying one gene is capable of producing only one polypeptide, not the entire protein. Okay, so this is the structure of the entire protein, hemoglobin. It is made up of 4 polypeptides. But the second hypothesis is saying only a single polypeptide can be produced by one gene. So the second hypothesis is one gene, one polypeptide hypothesis. The first hypothesis is one gene, one enzyme hypothesis. That is, the entire enzyme, the entire protein is produced. Whereas in the second hypothesis, one gene is capable of producing only a single polypeptide. It can produce only beta-2. It can produce only beta-2 polypeptide. Okay. So the two hypotheses regarding the gene, how whether it is capable of producing an entire protein or it is capable of producing a particular polypeptide. Okay. Now, in search of genetic materials, how genetic material is found out, the history of invention of the genetic materials, how DNA is found. First, a scientist named Wilhelm Hofmeister, he only separated the rod-like bodies which are found in the cell during the time of cell division, and he isolated the rod-like bodies during cell division and he named it as chromosomes. Okay, first chromosomes, the rod-like bodies which are formed during cell division is isolated by Wilhelm Hofmeister. And the second scientist, Friedrich Miescher, and he extracted a substance from the nucleic acids. He extracted from the nucleus and he named it as nuclein. So that is the genetic material. And he is saying the nuclein is the genetic material. So the first scientist, he called it as chromosome, and the second scientist, and he named it as nuclein. He extracted a substance which is from the nucleus and named it as nuclein. And the same substance is named by Altman. The third scientist, Altman, named the genetic material as nucleic acids. And after that, it was named as DNA. Okay, so this is the history of the invention of the genetic material. First, it was named as chromosomes, and then nuclein, and then nucleic acids, and now DNA. Okay, actually these chromosomes are made of protein and DNA. Chromosomes are made of protein and DNA. And another one experiment is that Griffith experiment. The name of the scientist is Griffith. He did an experiment to prove that DNA is the genetic material. So by the process of transformation is actually the exchange of genetic material from one bacteria to another. So that is transformation. Now that is otherwise conjugation. Conjugation, it is a type of reproduction. Two bacteria, they come close. This is one bacteria and this is one bacteria. They two come closer, and from one bacteria the DNA will be inserted to the nearby bacteria. So by this way it can transfer the nucleus and it transfer the DNA from one bacteria to another. This is bacterial transformation. During bacterial transformation, what is transferred? DNA is transferred. So the experiment is to prove the transferred material is DNA. So what is transferred? Okay, now we are saying during bacterial transformation DNA is transferred. So the same thing is proved experimentally by Griffith. So it is a name of the scientist Griffith. he did an experiment on mice. Okay, the experimental animal is actually mice. He used two different types of bacteria, two different strains of bacteria. One is rough strain and another one is smooth strain. Rough strains means the surface of the bacteria will be rough. Smooth strains, the surface of the bacteria will be smooth. Okay, the rough strain is non-virulent. Smooth strain is virulent. Virulent means it is capable of killing the mice. Virulent means some toxicity is there. It is capable of this bacteria will kill. So that is virulent. Invirulent means, so non-virulent means it will not cause death. Okay, it is non-toxic. So rough strain is no problem. If rough strain of bacteria is injected into the mice lives. The mouse is alive. There is no death. But smooth strain of bacteria is injected, the mouse dies because the smooth strain is virulent. Smooth strain is virulent. Okay. Virulent means some toxicity is there, it is capable of killing. And the third type, look at it here, the mouse is injected with heat killed smooth strain. Smooth strain is actually virulent. Smooth strain will kill, but here the smooth strain is killed by heat. That is, heat killed smooth strain is injected. When it is injected, the mouse is alive. Okay. If it is a normal smooth strain, the mouse dies. The heat killed smooth strain, mouse is alive. Okay, it is not capable of, it is incapable of causing death. Okay, and the fourth round, here the mixture of heat killed smooth strain and also rough strain. Rough strain is also non-virulent. Heat killed smooth strain is also non-virulent. This is also not capable of causing death. Okay. Here the mixture of rough strain and heat killed, both are non-virulent, but they cause death. How? Here the transformation takes place. Actually, in the DNA from heat killed smooth strain, the DNA from heat killed smooth strain is transferred to the rough strain, and it makes the rough strain to smooth strain. Once the DNA is enter into the rough strain is changed into smooth strain. You know, smooth strain is actually virulent. As a result, the mouse dies. So here, so this is rough strain and this is rough strain and this is smooth strain. From the smooth strain, the DNA is entering into the rough strain and it makes the rough strain bacteria into smooth strain. When it is becoming smooth strain, it will become virulent and it will cause death of the mouse. So from this we can understand DNA only transferred from one bacteria to another during transformation. Okay. So this is also another one experiment to prove that DNA is the genetic material. DNA only the genetic material. During transformation, DNA only transferring from one bacteria to another. Okay. Now, for this experiment, they are taking heat killed S bacteria. Heat killed X bacteria. So S strain is actually virulent, but it is heat killed. Heat killed means it is unable to kill. It will become non-virulent. So in this test tube, what is there? The liquid contains the mixture of all the nutrients which are there in the bacteria. The heat killed bacteria is ground and the extract is extracted from this bacteria. So what are the components of the extract? Look at that: proteins, RNA and DNA, lipids, sugars, everything will be there. The bacteria contains what and all? Bacteria contains proteins, RNA, DNA, lipids, sugar, everything will be there. So what is there in the first test tube? The test tube contains the extract of heat killed S bacteria, S strain of bacteria. So the bacteria contains lipids, sugars, proteins, RNA, DNA, everything is there. But what and all taken out? Now look at the first test tube. What is taken out? Lipid is taken out, sugar is taken out, and what and all remaining there in the mixture? Proteins will be there, RNA will be there, DNA is there. So the things which are removed from the extract is only lipid and sugar. So apart from that, what and all there in the mixture? Protein, RNA, DNA. Okay, and then this liquid is taken to these two test tubes. Okay, in these three test tubes, they are taking the sample from the above test tube, and in each test tube they are adding enzymes. The first two test tubes they are adding proteinase enzyme. In the second test tube they are adding RNase enzyme. The third test tube they are adding DNase enzyme. All these are enzymes. DNase, okay, RNase, proteinase, all these are enzymes. Once proteinase is added to this mixture, this proteinase enzyme will destroy the protein. As a result, there will be no protein. Okay, so in the test tube, no protein, because the protein is destroyed by the enzyme proteinase. The second test tube, what is the enzyme added? RNase. So this enzyme will destroy RNA. As a result, there is no RNA. In this enzyme, DNase enzyme will destroy DNA. As a result, the mixture, what will be absent? No DNA. There is no DNA. So here no protein, here no DNA, sorry, no RNA, and here no DNA. Okay. Now these samples are taken in the conical flask. Okay. In the conical flask, they are adding R strain of bacteria. Again, they are adding R strain of bacteria in each conical flask. Okay, in the blue color bacteria is actually R strain, that is rough strain. Okay, initially we take only S strain. Now only we are adding R strain to the each conical flask. Once it is added to the conical flask, what must be taken place? Bacterial transformation must be takes place. So there are two types of bacteria. First it was S cells, now it is R cells. Okay, now two different strains of bacteria are there in the conical flask. Now bacterial transformation must be takes place. DNA must be transferred from one bacteria to other. Okay, so here transformation takes place. As a result, S cells appear. Okay, once R cells are introduced, S cells appear. Here also S cells appear. Here also transformation takes place. But here there is no S cells. S cells are not appear. Okay, the reason is there is no transformation takes place. Why transformation is not takes place? Because there is no DNA. Since there is no DNA, there is no transformation. Okay, so for transformation, what is required? DNA is required. So during transformation, DNA only transferred from one bacterial cell to another. So from this experiment also we can understand that DNA is the genetic material. Okay, now we are testing, in this experiment we are testing three materials which may be the genetic material, whether it is protein or it is RNA or DNA. Okay, that is the experiment. So at the end of the experiment we are coming to the conclusion which only must be the genetic material. The DNA must be the genetic material. Okay, okay, because in this sample there is no DNA. Since there is no DNA, there is no transformation takes place. Okay, for in this conical flask, first two flasks transformation takes place. Okay, because DNA is there. In the third test tube only there is no DNA, as a result there is no transformation. So from this we can understand transformation requires DNA, and DNA is the genetic material. DNA is the genetic material. Okay, with that we can finish this lesson.